Director, Amity School of Hospitality, Dr. Sanjeev Pal, sir. Deputy Director, Amity Institute of Microbial Technology, Dr. Sanket Joshi, sir. And all my dear students, I welcome you to the first technical session of the third track of Sustainable Development in Applied Science, Engineering and Technology for the International Convention on Green Environment, Technology, Entrepreneurship through Innovation. Dr. Sanket Joshi is currently serving as Professor and Deputy Director at Amity Institute of Microbial Technology, Amity University, Rajasthan. Sir has served as Deputy Director, Oil and Gas Research Center, and an application specialist at Sultan Qaboos University, Oman. Professor Joshi has an experience of 19 years of academic teaching and research, and 4 years of industrial research and development experience in India and Oman. While working in Indian pharma companies, he undertook several turnkey projects on antibiotics, anti-diabetic drugs, and coenzyme Q10. His current research interest encompasses energy, microbial products, and environmental bioremediation. He has 140 publications as papers in international journals, book chapters, international books, 1 Australian innovation patent, and conference abstracts to his credit. He serves as the editorial board member for some of the highly reputed journals. He has an H index of 5041, citations of 5041, an H index of 33, and an ITIN index of 75. Sir has been listed among the 2% of the Stanford University Elsewhere list of highly cited scientists in the world for the year 2022 and 23. For excellence in scientific research in energy and biotechnology disciplines. With these few words, I would like to welcome Professor Sanket Joshi, sir, for the welcome address. Sir, please.
Thank you, Dr. Manishita. So I will not take much of your time. Please settle down, settle down. थोड़ा बैठोगे तो जो खाना खा के है वो सेट होगा थोड़ा पेट में, तो थोड़ा ज्ञान भी जाएगा. Your blood flow will start moving from your stomach to your brain.
क्या करना है? एक सेकंड जाओ. ये वाला ना. अब आप यूज कर सकते हो. हैं?
So good afternoon to all. Without taking much time, आ जाओ बैठ जाओ जो लोग पीछे हैं. Already थोड़ा late हो गया हमारा. So let's start with the conference, this session. So I think all those guys who are sitting here, they know this session or this particular conference is mainly dealing with science and technology and with innovation, which is the motto of Amity University, Rajasthan. So we are lucky enough to have 2 young achievers amongst us. One is a female representative and another one is a guy. So these 2 young achievers have done contribution in different fields. One is a professor in biotechnology, Professor Nilima. And another person, Dr. Gaurav, he is an expert. He is serving in Russia also, and he is an expert from engineering field who is working on energy. So the first speaker will be Professor Nilima Kumari. So just to introduce her with few words. She is a professor in the Department of Bioscience and Biotechnology, Banasthali Vidyapith, Rajasthan. She obtained her biotechnology doctorate in year 2005, and her doctoral and postdoctoral visits and famous institutes in Germany as well as in Finland. Both the countries are doing quite good in biotechnology. And she has an impeccable track record in academics with a standing of around 25 years of service at Banasthali Vidyapith. She is currently working in the field of plant biotechnology, and she has made significant contributions in plant stress biology, which is globally acclaimed. She has published more than 75 research papers in highly reputed journals. She has also guided more than 23 PhD students now, and many of whom are already well placed in different academic institutes as well as industries. She has also been conferred with 2 Young Scientist Awards, 1 Women Scientist Award medal, and 1 Distinguished Scientist Award by reputed national scientific societies. So this is just to give an overview. Please welcome, ma'am. करते करते आपका यहाँ पे यहाँ से भी हो जाएगा, कोई दिक्कत नहीं। मैम मैम बोल रहे हैं इफ इट इज शी इज फाइन इफ इट वर्क्स फ्रॉम हियर, तो उनका पीपीटी ओपन करना पड़ेगा। इनका डेस्कटॉप पे सेव किया है शायद। हाँ ये है, एक सेकंड। हाँ हो जाए।
शेयर भी कर रही हूँ, शेयर करना पड़ेगा। I hope I'm audible. हाँ वह तो नहीं है तो कोई बात नहीं, चलेगा। देखती हूँ Respected and learned faculty members of MIT University, Rajasthan, eminent speakers of the conference, participants of the conference, and dear students, a very good afternoon to all of you. First of all, I extend my sincere thanks to the higher authorities of MIT University
and the entire team of this international convention for inviting me as keynote speaker in this conference. I shall speak on nanoparticles for enhancing agricultural productivity under abiotic stress conditions. The abiotic stress conditions include the extremes of temperatures, both cold as well as heat, the scarcity and the excess of water, means the drought and flooding. Then it also includes salinity, alkalinity, and also the heavy metal stress prevailing mainly in the soil as well as in water. The abiotic stress factors, they directly affect the growth and productivity of the plants. So for example, the cold or the freezing stress will cause ice crystal formation, which will damage the cell, and thereby it will create disorders in the metabolism of the plants. The heat stress will definitely reduce the photosynthetic efficiency of the plants as well as the transpiration efficiency. It affects the water use efficiency of the plant, and ultimately this will affect the productivity or the yield of the plants. The heavy metal stress will also disturb the metabolism of the plant, thereby overproducing the reactive oxygen species. It will also lead to, you can just switch it on. It's on. It will cause the accumulation of the heavy metals, and this will also lead to the damage to the DNA or the proteins and carbohydrates in the cell. Yeah, the drought stress may cause the stomatal closure, and it will also again inhibit the photosynthetic machinery in the plants, and it will also inhibit the transpiration because these activities are directly related with the water transport and the utilization of water. Similarly, the flood stress will cause hypoxia or anoxia, which will disturb the you can say respiratory chain and lead to disturbance in the ATP synthesis in the plants. Then the salinity stress will also lead to membrane damage and the nutrient imbalance in the plant. Besides, the photosynthetic activity is also disturbed. Now, whenever the plants are exposed to any environmental stress, this will lead to disturbance in the overall metabolism of the plant, which will reduce the productivity of the plant. It will reduce the growth and development of the plant, which will be reflected through various biochemical, physiological, and molecular levels. Now, the plants will respond to this stress and it will try to overcome these stress conditions by showing various tolerance mechanisms. Now, nowadays we see that the population of the world is increasing day by day, and there is a dire need to boost the production of the food crops, and that has to be through the sustainable methods. The agricultural productivity is reduced due to both biotic as well as abiotic stress conditions, and this reduction in the productivity is generally 65 to 87%, which is quite a bit. And mainly the abiotic stress factors, they are responsible for roughly half of the yield losses. Now, the plants will show several strategies in order to cope up with the abiotic stress. So, in our lab, we are working on in plant stress biology. We are dealing with several plant systems and also different types of abiotic stress. So, what we do is we expose the plants to different type of abiotic stress, and then we will analyze the biochemical, physiological, and molecular parameters as well as the gene expression in the plant, which will be directly affected by those stress conditions. So, and then we will provide some alleviation strategy so that the plant is able to overcome that stress condition. So, for example, some of the strategies which we are working with is the application of the nanoparticles, then the application of the phytohormones, for example, like salicylic acid, methyl jasponate, etc. Then the application of metal-allyl combinations like aluminium and fluoride, then silicon and fluoride, etc. And the application of the arbuscular mycorrhiza, which will help the plant to overcome the stress conditions. So now whenever the plant will be under some type of stress condition, it will show reduced productivity, and through these strategies, the plant will show abiotic stress tolerance and it will try to overcome and come out of that stress condition. So which is reflected through all of these. So today I shall focus on one of these aspects, that is the application of nanoparticles, and currently 4 of my students, they are working in this area. So I hope everybody knows nanotechnology. So this nanotechnology is dealing with the nanomaterials which are of the scale that is 10 to the power minus 9 meters. These days, there are various nanomaterial containing products in the market, including the textiles, pharmaceuticals, cosmetics, paints, in agriculture also. These nanoparticles may be of different types. They may be polymer based, they may be lipid based, or they may be non-polymeric based. These nanoparticles, they can be synthesized through different methods. These include the physical methods, chemical methods, and biological methods. Now, without criticizing the physical and the chemical methods, we prefer biological methods because these biological methods are eco-friendly, because we have to deal with the sustainable methods, or we have to carry out the sustainable development of the plant. So obviously these biological methods which are used for the synthesis of these nanoparticles, these are eco-friendly. When these nanoparticles are synthesized through the biological methods, then these have to be characterized in order to find out whether the process which we have carried out has led to the production of nanomaterials or the nanoparticles or not. So these nanoparticles, they can be characterized through highly sophisticated techniques, which includes transmission electron microscopy, scanning electron microscopy technique. It includes X-ray diffraction technology, then Fourier transform infrared spectroscopy, UV visible spectroscopy, dynamic light scattering also. So generally these spectrophotometric techniques are specially used because these nanoparticles, they show optical properties. So those properties are utilized in order to characterize them. So that means through these techniques, we are finding out whether the nanoparticle has been synthesized and it is of the scale of nanoparticle. So then only we can use them. Now these nanoparticles, they interact with different components. So these different components may be plants. That means the plant will be directly interacting with the nanoparticle. They may interact with the rhizosphere microflora because the bacteria, fungi, etc., they are present in the rhizosphere, which will interact with the nanoparticles, as well as the root exudates, which include some organic acids also. And these nanoparticles may also directly interact with the soil. So I'll give you just one example. For example, if it interacts with plants. So let us say if we spray these nanoparticles on the leaf. So the outer layer of the leaf has cuticle, and the size of the cuticle, the pore size, it will permit only particles which are less than only 5 micrometers, the 5 nanometers less. So that means even if you synthesize the nanoparticle, then only very, very small nanoparticles, they can enter the leaf and the cuticle will obstruct them. So this will be their interaction. It will not allow the nanoparticle to enter in the plant. But if we see the stomata, again, these are also present on the leaf, then the larger nanoparticles, they will enter the leaf through the stomata. So this is just one example to show how the nanoparticles will interact with the plant. Then the further barriers will be there and the interaction will be there. Similarly, it will interact with the rhizomicroflora and the soil components also. Now, these days, this nanotechnology is widely applied in agriculture. So this may lead to the improvement of the soil by providing the nutrients. So it will improve the nutrient efficiency in the soil, and these nutrients, they can be absorbed by the plants. This will lead to the crop production as well as the improvement of the crop, and this may also help in precision farming through the use of nanosensors, generally for monitoring the soil conditions. These nanoparticles are very, very advantageous. That is why they are being used. And one of the most important advantage of these, besides many, is that they increase the productivity of the plants and they improve the quality of the soil. These can be monitored smartly, and different techniques they can be used for all of these. Now, these nanoparticles, generally now they are being utilized in the form of nanofertilizers. So there are certain problems with the conventional fertilizers. The size of the conventional fertilizers is more than that of the nanoparticles. Now, when these conventional fertilizers are applied, these are very easily lost, or we can say that these leach off from the soil, or they run off the excess release of the nutrients, which leads to the toxicity and the imbalance in the soil. The conventional fertilizers, these have lower solubility. So that means there are issues with the bioavailability of the fertilizers because of the large size of the conventional fertilizers. So thereby the nutrient use efficiency of the plants will be low, or we say that the plants will not be able to so effectively use these conventional fertilizers, as sometimes they may also be converted into the insoluble form. So an alternative to these conventional fertilizers is the nanofertilizers or the nanoparticles in the form of fertilizers. So these nanofertilizers, they manage the soil health, they improve the seed germination and also enhance the growth and productivity of crops. They may also be used in the form of sensors to detect the heavy metals in the soils. They may be used as nanoremediators, so thereby they will act as sustainable mitigation of the organic pollutants, pesticides and heavy metals. These can also be used in the form of nanopesticides, so that means they will manage the different types of plant diseases and improve the plant health which the pest is causing otherwise. So obviously these nanomaterials, they have the advantage and they increase the stress tolerance in the plants. The same is depicted in this picture. Now these nanofertilizers, these are released in a controlled manner in the soil. So we say that there is reduced or undesirable loss of the nutrients from the soil. So that means they will be more available to the plants, which will improve their productivity. So that is what we call as nutrient use efficiency. These nanotechnology based agriculturally important nanofertilizers are being used currently because they have certain advantages. That is, they can form stable suspensions, their surface area will be more, and these are highly efficient. Now let us see the different methods of the delivery of the nanoparticles to the crop plants. So these can be applied through various measures, which include the coating of the seed, soil drenching. They can also be sprayed onto the leaves, that is known as foliar spray. They can also be added to the hydroponic systems. They can be directly injected into the roots. Then they can also be delivered through the biological vectors, and also the nanoparticle embedded fertilizers are there which are added to the soil. Now all these different types will be accessed differently by the plants. Out of all these methods, the two more common modes of application of nanoparticles are the foliar exposure, that means we spray the nanoparticles onto the leaves of the plants, or the root exposure, that means the nanoparticles will be added to the soil which will directly reach the roots. After the addition of these nanoparticles either to the leaves or to the soil, these are uptaken by the plant. So there will be a specific route of uptake of these nanoparticles in the plant, and then they will be translocated to different parts of the plant. So for example, if these nanoparticles are applied onto the leaves, they will enter through the leaf epidermis or through the stomata. They will reach the vascular tissue through the apoplastic or the symplastic pathways. The apoplastic pathway will include the cell wall, and the symplastic pathway will include the passage through the cytoplasm. Then after this, they reach the vascular tissue, that is the xylem and phloem, and from this they will be directed in both upward as well as in the downward direction, and they will virtually reach to all parts of the plant. Because if it is phloem, then they will travel downwards also. So it has been observed that if you spray the nanoparticles onto the leaves, they were found in all parts. This is indicating that these are translocated through xylem and phloem. So it's not only upward, but it is downward also. Now after this penetration, these nanoparticles are transferred, and then these are accumulated in different parts of the plant. And this will be affected by various abiotic stress conditions like humidity, light, temperature, salinity. And when all these abiotic stress factors will be there, then the plant's physiological characteristics, they will also affect the translocation of these nanoparticles. And this also depends on the properties of the nanoparticles. That means what is their size, what is their shape, what is their charge also. So for example, let us say if the nanoparticle is positively charged, so then the negatively charged, let us say for example, the organic acid, it will hold on the nanoparticle and it will not allow it to be accessed by the plant. So that is why all these properties of nanoparticles, they will also affect their accumulation or the accessibility. Now, let us see the effect of these nanoparticles on the plants. So broadly speaking, these nanoparticles will increase the activity of different types of enzymes in the soil. They also affect the photosynthetic efficiency of the plant. They directly affect the growth and development of the plant through nutrient uptake. And finally, we say that these nanoparticles affect the yield of the plant. So this yield of the plant will be directly reflected in the agricultural productivity. Let us say if we are concerned with the crops. So now there are different types of abiotic stress, and these nanoparticles, they can be applied under different type of stress conditions, which we already discussed. And in all the types of stress, these nanoparticles, they will try to alleviate that particular type of stress condition and enhance the, let us say, photosynthetic capacity. Okay, thereby it will enhance the growth and development of the plant. That means it will affect all those molecular mechanisms and finally increase the productivity. So for example, these nanoparticles, they may be applied to the plants in case of drought stress. These have been reported in several plants, including soybean. These nanoparticles can be applied under the salinity conditions. And as I said that when these nanoparticles are applied, then they will act and they will cause different types of metabolic and biochemical changes, which will enhance the growth and productivity of the plant. So let us say they will balance the, or they will cause the ionic homeostasis in the cell. They will optimize the nutrient uptake. Then they will reduce the reactive oxygen species which harm the cells. They will enhance or increase the photosynthetic efficiency of the plants. They will modify the plant hormonal signaling system. They will modulate certain gene expressions, and they will also lead to production of, let us say, nitric oxide. Similarly, in the heat stress also, when the nanoparticles are applied, the plants will show different types of stress responses, and finally the productivity of the plant will be increased. The same is for the heavy metal stress also. So that means through these slides, I'm just trying to say that whatever is the type of stress, these nanoparticles can be applied to the plants which are exposed to different types of stress conditions. So those stress conditions will be reflected by the change in certain parameters. They will reduce the productivity of the plant, but when these nanoparticles are applied, they will enhance the tolerance capacity of the plant, and it will help the plant to cope up with the stress condition and increase the productivity of the plant. One of the mechanisms is the effect on the antioxidant defense mechanism also, which includes both the enzymatic as well as the non-enzymatic machinery. This is also promoted. The stress also affects the electron transport chain in mitochondria, because electron transport chain is directly linked with the ATP synthesis in the plants. And when this chain is affected, then it will also lead to the production of the reactive oxygen species. So these reactive oxygen species, they are very harmful for the cell. This stress also affects mainly the photosynthetic apparatus. So we say that photosynthesis is one of the primary processes which is affected by the plant under any stress condition. So thereby overall the yield of the plant will be affected. Now, through the use of these nanoparticles, all these different types of stress may be alleviated. So this is the combined diagram in which we say that different types of nanoparticles, they can be synthesized. For example, zinc oxide nanoparticles, silicon dioxide nanoparticles, gold particles, silicon particles, silver particles, manganese oxide, iron particles, hematite, and many others. So different types of nanoparticles, in fact, this titanium oxide nanoparticles, they can also be synthesized. Magnesium oxide is there, then copper nanoparticles are also there. So one of the strategy is what type of the nanoparticle is to be used. The another is, be it any type of stress, it will affect the plant. So when these nanoparticles are delivered to the plant through some strategy, which we listed, okay, and the main one was the foliar spray or through the root exposure, then this nanoparticle exposure will improve the antioxidant defense system. It will improve the plant growth and germination. It will lead to increase in the osmolytes. it will reduce the ionic toxicity in the plants. It will improve the plant-water relations, thereby increasing the water use efficiency. The signaling pathways will be activated. There will be efficient nutrient accumulation in the plant. There will be detoxification of the reactive oxygen species through the antioxidant machinery. So overall, all these processes will enhance the productivity of the plant. Now, such type of studies have been recently carried out in several plants. For example, in wheat, maize, then in onion, then rice and capsicum. So in all of them, a different type of nanoparticle is applied when the plant is under certain condition, and the plant will come out of that stress condition when the nanoparticles are applied. So for example, these magnesium oxide nanoparticles, they were synthesized and it affects the seed germination in Arachis hypogaea. In another example, copper oxide nanoparticles, they affect the growth and physiology as well as nutrient uptake and cadmium accumulation. So that means now it affects the plant under the cadmium stress or the heavy metal stress. These experiments have been carried out in wheat. Zinc oxide nanoparticles, they have been shown to enhance the drought tolerance in wheat also. So I shall focus on one example, though we are working on different plants. Okay, I told you that 4 of my students are working in this area. So I will just explain all this with the help of one example. So the example which I have selected is the heavy metal stress in the plant. So when the soil is contaminated by heavy metal, then it will cause or impair the growth of the plant. Now, according to the Agency for Toxic Substances and Disease Registry, the arsenic tops the list in the list of the toxic substances. That means it is the most toxic substance. In our lab, we have carried out work on arsenic, lead and mercury. That is number 1, number 2 and number 7. So I will take the example of arsenic which tops the list. So this arsenic, this is very, very harmful, and according to World Health Organization, the permissible limit of arsenic in the groundwater is just 10 microgram per liter because it is highly toxic, you see. And in the soil, it's the toxic concentration is if it is more than 20 milligram per kilogram. This arsenic occurs in 2 forms, that is arsenite and arsenate, according to valency 3 and 5. So out of both of these forms, the arsenic 3 is highly toxic, and this has effect on the plants. And this arsenic contamination, this will affect the quality of the food in the crops. And this will also enter the food chain since these crops will be consumed by humans and also they can be consumed by the animals. The source of the arsenic may be anthropogenic, that is through the humans, or they may be geogenic. The geogenic sources include the weathering of the rocks and the volcanic eruption, which add the arsenic to the surface water, which reaches the groundwater. The anthropogenic sources, they include various agricultural practices like the use of fertilizers, pesticides, etc. Industries may also add to the arsenic, and there may be other methods like the sewage is there, then the municipal waste, treatment of the municipal waste is also there, and the smelting also. So that also adds arsenic to the water. Now, nearly 107 countries in the world are affected by arsenic contamination. So that means this is a this is a problem for everybody. So this is very, very important. So here the groundwater is below the permissible limits, that is 10 ppb, parts per billion. So here in this graph, in this diagram, in this map, we have shown the countries in which the arsenic pollution is there in the groundwater. So that means this becomes very, very relevant since this will reach the food chain. Now let us see the effect of the arsenic on the plants. So arsenic reduces the germination percentage. It reduces the shoot and root growth, the number of the leaves and leaf area. There is decreased plant biomass, so the yield of the crops is affected. Mainly, the photosynthetic machinery of the plant is affected, so thereby the rate of the photosynthesis will be decreased. It will lead to production of reactive oxygen species, which will damage the cell, and it will also alter many enzyme activities involved in this enzymatic as well as non-enzymatic machinery. And it will also increase the lipid peroxidation. So overall, we say we can summarize that when the plant is exposed to arsenic, it will affect the growth and productivity of the plant. Now here the nanoparticles may be applied so that the plants are able to overcome the heavy metal stress. So obviously the nanoparticles will interact with the soil, and these heavy metals, they will minimize the mobility and the bioavailability of the nanoparticles. The nanoparticles will help in alleviation of these heavy metals by modifying the heavy metal transporters. So that means if it blocks the heavy metal transporter, then the heavy metal will not be able to enter the plant and harm it. Then these nanoparticles, they may also activate the defense system of the plant, which includes the antioxidant machinery. Here we have synthesized the zinc oxide nanoparticles. So we propose that these zinc oxide nanoparticles, they are a unique stress mitigator, and they have the potential to increase the growth of mung bean plants under arsenic stress. So this mung bean plant is very, very important economically. And since this is a rich source of protein, carbohydrate, vitamin, etc., and mainly it is consumed in the form of sprouts and dal. Now, this crop is very, very important because India is the largest producer of mung bean, and it produces nearly 65% of the mung bean on the world scale. So the plant which we have chosen is mung bean, which is a very economically important plant. Then the nanoparticle which we selected was zinc oxide. So we selected or we synthesized the zinc oxide nanoparticles, obviously through the biological methods. The zinc oxide nanoparticles were synthesized using the extract of another plant, that is Emblica officinalis. So the fruits of Emblica officinalis, that is amla, they were used to synthesize the nanoparticles. So that is why we are saying that these are biosynthesized. So they will be eco-friendly since they have been synthesized from a biological source, and obviously one of the plants which is very, very important, that is amla. So they cannot be harmful, we can say that. Now this scheme shows the synthesis of the zinc oxide nanoparticles. So first of all, the zinc acetate solution is taken, which is mixed with the fruit extract of the amla, and after this they are mixed, and which will lead to the change in the color, and finally after drying we get the this powder of the zinc oxide. Now next step will be, we have to find out whether this powder, it contains nanoparticles or not. So that means we have to characterize them. So in order to characterize these nanoparticles, different techniques were used. For example, UV visible spectrophotometer, and through this it was found that the peak was observed at 345 nanometers, which confirms that the zinc oxide nanoparticles have been synthesized, because this peak will be specific for zinc oxide. The other techniques which were used were X-ray diffraction technology, and through that X-ray diffraction technology, we confirmed that the nanoparticles which were synthesized, their size was around 24 nanometers. Then the zeta sizer and potential were also used in order to determine the size and stability of the nanoparticles. So nanoparticles of around 28 nanometers were synthesized, which were quite stable. Through the electron microscopic techniques and through the EDX, we determined the morphology of the zinc oxide nanoparticles. So it was observed that the nanoparticles which were synthesized were spherical in shape. And through this EDX, it was confirmed that zinc and oxygen is present in the nanoparticle, and that their ratio indicated that the zinc oxide nanoparticle have been synthesized. Then the functional groups were also detected by Fourier transform infrared spectroscopy, and through this experiment the conclusion was again that the zinc oxide nanoparticles were synthesized. Now, the experimental setup was carried out in which the plants were exposed to different concentrations of arsenic under different stages, that is under the vegetative stage and reproductive stage. So obviously when the plant is exposed to arsenic, then it will lead to reduction in the growth of the plant. So 5 varieties were taken, And in these 5 varieties, in all the 5 varieties, with increase in the concentration of the arsenic, decrease in the growth of the plant was observed. Now what we had done is we had selected 2 varieties, one was tolerant and the other was sensitive, and further experiments were carried out on these 2 varieties. 2 stages were decided for the experiment, that is the vegetative stage and the reproductive stage, that is at 25 days and 45 days after sowing. I will just summarize now what was done in the experiment. So when the particular concentration of nanoparticles was applied, it was observed that they affect the growth parameters in the plants. For example, the shoot length, root length, their shoot dry weight, then root dry weight, the leaf area, as well as the number is affected. It was increased. So that means the spraying of the nanoparticles, it helped in increase in the growth and development of the plant, which was reflected through these parameters. Now, when these nanoparticles were applied, then different types of physiological, biochemical and molecular effects were also observed. For example, the effect on photosynthesis, then the reaction in oxygen species and other enzymatic machinery was observed, and certain gene expression studies were also carried out. Now, in all the studies, when these growth parameters were observed, so for example, when the zinc oxide nanoparticles were added, then the shoot length, root length, leaf area, all these were increased under the arsenic stress also. That means if you apply only the nanoparticle, then also it increases the growth of the plant. And if you apply the nanoparticle after the application of the heavy metal, which had otherwise reduced the growth of the plant, then also it enhances. That means now these nanoparticles are helping the plant to cope up with the arsenic stress and increasing its productivity. So these experiments confirm the same in 2 varieties. Besides these growth parameters, the photosynthetic performance of the plants was also observed. The photosynthetic pigment content under the arsenic stress was reduced, but when the zinc oxide nanoparticles were applied, then it led to increase in the photosynthetic pigments, and obviously the increase in the photosynthetic pigments will help the plant to photosynthesize better. These experiments were carried out with several concentrations of zinc oxide, but we observed that if we increase the concentration of nanoparticles also, then they are also inhibitory for the growth of the plant. That means we cannot increase the concentration of nanoparticle unendedly. We have to choose a particular concentration which will enhance or which will increase the growth and productivity of the plant. So we have to decide it through different experiments, taking different concentrations of zinc oxide. Then after these photosynthetic pigments, the stomatal opening size was also observed. So it was observed that a concentration of 20 milligram per liter of zinc oxide nanoparticles, it maintained the structure of the stomata. So that means if the stomata are not damaged or deformed, then they will allow the smooth functioning of the stomata, thereby the photosynthesis also. So these were the studies. So in the arsenic contaminated, means in the plants where the arsenic was given, then the stomata closure was there. But when the nanoparticles were added, the stomata were open. The photosynthetic measurements were carried out through dual PEM. So in which we measure the photosynthetic efficiency of both the photosystem 1 as well as photosystem 2. So this instrument is known as dual for both, okay? And PEM means pulse amplitude modulated fluorometer. So this instrument measures the fluorescence in the leaves and then it converts it into the photosynthetic parameters. That means it will measure the electron transport rate, okay? What is the electron transport rate in the plant? What is the photosynthetically active radiation and others? So here it was observed that when the plant was exposed to arsenic stress and the zinc oxide nanoparticles were applied, then it led to increase in the photosynthetic efficiency of the plant, which was reflected through the photosynthetic parameters. For example, let us say ETR max, that means maximum electron transport rate, then saturating photosynthetic photon flux density and other parameters. These graphs, they represent the photosynthetic, different types of photosynthetic parameters when the plant was exposed. Then the zinc oxide nanoparticles, they also affected the antioxidant enzyme activity under the arsenic stress in these plants. So here it was observed that the application of nanoparticles will enhance the antioxidant machinery, that is the activity of the different enzymes like ascorbate peroxidase, then catalase, peroxidase, superoxide dismutase, glutathione reductase, glutathione S transferase, and non-enzymatic antioxidants like polyphenols, flavonoids, etc., they were also increased. So all of these will protect the plant and they will reduce the reactive oxygen species which otherwise harm the plant. The flavonoid content was also increased and the polyphenol also. The proline content was also increased. The MDA content was also increased as well as the content of hydrogen peroxide was also increased. Finally, we also measured the gene expression of PSBA. This PSBA gene codes for the D1 protein, which is one of the major protein which is present in the photosystem 2 of the plants. So that means directly it is measuring the efficiency of the photosystem 2, where we say that the photolysis of water takes place and the electron transport starts from P680 or photosystem 2. So it was observed that it led to change in the or increase in the gene expression, thereby protecting the photosystem 2. Then the arsenic and arsenide, they were also, their accumulation was also observed, and it was observed that when the nanoparticles were sprayed, then there was less accumulation of arsenic in the plant tissues. So that means they were holding them below, okay, they were not allowing the arsenic to enter the plant. So this was in different Finally, we concluded that the application of the zinc oxide nanoparticles, it modulated the or it helped the plant to cope up with the arsenic toxicity by decreasing the arsenic concentration in different parts of the plant. The zinc oxide nanoparticles, they promoted the growth parameters, different types of biochemical response, antioxidant activity, photosynthetic parameters, etc. And a concentration of 20 milligram per liter of nanoparticles, that was important to control this toxicity. So that means the conclusion is that these zinc oxide nanoparticles, they can be used as a sustainable approach in order to alleviate the arsenic stress in mung bean plants. So now this study was published in Archives of Agronomy and Soil Science. Another paper was in Journal of Soil Science and Plant Nutrition, where we propose that these zinc oxide nanoparticles, they act as a new fertilizer and they will help the mung bean plants to cope up with the arsenic stress. One of the papers was published in Vegetos and another was published in Reviews of Environmental Contamination and Toxicity. So where it was proposed, the green agriculture was proposed, that is these nanoparticles, they can be used as tools to mitigate or to alleviate the heavy metal toxicity. So similar type of studies were carried in another plants. I will not elaborate on this. So iron nanoparticles were synthesized and their effect was observed on Trigonella phoenum graecum, that is methi, and it was exposed to drought conditions. The plant growth, yield, photosynthesis as well as the nitrogen metabolism was observed, and it was observed that these nanoparticles are able to alleviate the drought stress in the Trigonella. This study was published in 2 papers in plant stress. So both these papers, in one paper the photosynthetic performance was shown and in another paper the effect on the nitrogen metabolism was observed. So that means it increased the biological nitrogen fixation efficiency of the plant. In third study, the silicon nanoparticles were synthesized and these were sprayed on oat plants, that is on the avena, and the plant were exposed to fluoride stress. So finally it was observed that these silicon nanoparticles, they were able to alleviate the fluoride stress in oat plants. In one of the studies, magnetite, that is iron nanoparticles, have been synthesized from lemongrass, that is Cymbopogon citratus. The effect of arsenic stress was observed on Vigna mungo, that is black udder, and it was observed that these magnetite nanoparticles, they help the plant to cope up with the arsenic stress. So in conclusion, we say that these nanoparticles in general are beneficial for the plant and these act as nanofertilizers and they increase the productivity of the plant. However, there may be certain limitations because they may cause some chemical hazards to the edible plant, so they should not enter the food chain. Then sometimes they also generate free radicals. It all depends on how they are interacting with the different components, which I told you initially. Sometimes they may not be stable and they may be toxic, and the energy consumption and the cost of production is also important. Many thanks to Professor Vinay Sharma, Dean Research and Director PhD Program and Director MIT University of Biotechnology, MIT University Rajasthan. I am fortunate to be his student, and he has been providing his expert guidance to us for all these studies. I also acknowledge my students, Anju Sogarwal, Sushma Rani, Shweta Bish, Sonal Mishra, Gargi, and Sahib Kaur, who carried out these experiments. I acknowledge my university, Banasli Vidyapeeth, Rajasthan, to provide all the facilities for conducting these experiments. The facilities provided by Department of Science and Technology, Government of India under DST Curie and DST FIST program is also acknowledged, and the CSIR New Delhi is also acknowledged for providing the fellowship to my students. Thank you. Anyway, this is an emerging field, so we have to try how successful are these nanoparticles in the agriculture. Thank you all.
On behalf of the organizing committee, I would like to thank ma'am for sparing her valuable time and giving us an insight on such an important upcoming area of the field. Thank you so much, ma'am, for sparing your time and focusing on this important aspect. And I hope our students have learned a lot from this. Moving ahead with the program. Our second keynote speaker for the day, Dr. Gaurav Pandey, sir, is the chief scientist in Russia and assistant professor at University of Petroleum and Energy Studies, UP-ES Dehradun. He is a distinguished figure in the realm of scientific research and academia. His expertise spans across advanced energy systems, environmental technology, and sustainable development. At UP-ES Dehradun, he contributes significantly to the academic community, mentoring students and leading research initiatives that address critical global challenges. His role as chief scientist in Russia further underscores his commitment to pioneering research focusing on innovative solutions for energy efficiency and environmental conservation. Sir's work is characterized by a multidisciplinary approach, bridging the gap between theoretical research and practical applications. His contributions have not only advanced scientific knowledge, but have also fostered international collaboration, reflecting his dedication to addressing global energy and environmental issues through research and education. With these few words, I would like to hand over the dais to Sir. Welcome, sir. Thank you for sparing your time.
Am I audible? I hope all of you have had lunch, a good had lunch, right? Not feeling sleepy? I will not take much time. Can you raise your hand for those who are from engineering background? Okay, fine. And those who are from science background? I can see the common hands. Okay, fine. presentation open up. Video चलेगा ना?
Share it with the group.
हाँ?
I have to share it online as well because this is hybrid model.
Okay, fine. So this is new topic. So I will try to discuss with all of you in layman language. In case if you are not able to understand, please raise your hand at any point of time. Okay. Feel comfortable. Thank you, ma'am, for introducing me. And thank you for Professor Sanket Joshi for inviting me here. And I would like to thank you to all of the participants, delegates, and audience and students, those who are going to attend my talk. So I'm going to talk on the energy storage part. So my topic is energy horizon. So I will discuss about the cutting edge or the innovative technologies of the gas hydrate systems for the sustainable future. So I work as assistant professor in UPES, and I'm also a chief scientist in Russia, Earth Cryosphere Institute, Tyumen, which is located in the Siberia part of the Russia, right? So this is my academic journey. I did my BTech, Master's, MTech in petroleum engineering, then completed my PhD jointly with IIT Madras and NUS Singapore. Some of my projects achievements, some of my awards achievements, and I'm also serving as professional activities as a reviewer, session chairs at different level. So I'm not going to take much of time to discuss this. My PhD supervisor, Professor Praveen Linga at NUS Singapore and Professor Jiten Sangwai at IIT Madras. Both are in chemical engineering department at their respective institutes. So as we know that we are worrying about the climate change, right? So we are focusing on the carbon reduction or reducing the carbon emission. So if we look at the data here, the global CO2 emissions, which is generated by the industrial processes and the combustion processes, if we look at the China is releasing 28%. And if we look at the US, which is releasing 15%, and India emits only 2.65 gigaton CO2 annually, which is 7% of the world carbon emissions. And if we look at the annual increment here, emissions, which is 2.19 gigaton. So still there's a challenge or topic we have to worry about how to reduce this carbon emission or how to capture, how to store for the longer period of time this CO2. So there are several technologies we are following, but we have to look at the carbon footprint, how we can tackle these situations or tackle these problems. So as moving towards the carbon reduction emissions, we are focusing on the decarbonization. What is decarbonization? Reducing the carbon emissions, right? But still decarbonization cannot help you. We have to move towards the clean energy. If we talk about clean energy, we can depend on the hydrogen because hydrogen is only the cleanest source of energy. But achieving the decarbonization, we have 2 or 3 approaches, holistic approaches, managing, and also we have to look at the economic part of the system. So energy transition, we are moving from the fossil fuels to another energy resources like solar, wind, nuclear, etc. But all these factors depend on the renewable energy, energy efficiency, sustainability. So there's a need of energy transition because we are not able to solve the carbon problem. So either we shift towards the completely towards the clean energy or we can reduce the carbon emission. So I will change a little bit this topic in another point. We have to look at the energy recovery, energy storage. Then we will come back again to the same topic, how to achieve the carbon reduction of the carbon emission or how we can achieve this CO2 problem. So according to International Energy Agency, this era is called as golden era for natural gas. So if we look at the global scenario, till we have this map for 2035, which represent the shares of natural gas is highly significant. And another, if we look at the Singapore scenario, the share of natural gas is highly significant, as we completely depend how we can solve this problem regarding the natural gas, because we are totally dependent on the natural gas, and natural gas also release the carbon emission. What is natural gas? Can anyone tell me? What is natural gas? अरे जो भी पता है वो बोल सकते हो, ऐसा कुछ नहीं है कि बिल्कुल एग्जैक्ट आंसर देना है. आर यू अवर विद द एलएनजी एंड सीएनजी? है ना, सीएनजी सुना है, एलएनजी भी सुना है. ऐसे ही नेचुरल गैस होता है, ये नेचुरल गैस के बाई प्रोडक्ट्स हैं, राइट. सो द नेचुरल गैस विच इन विच प्राइमरली कंसिस्ट ऑफ मीथेन, जिसमें मीथेन का सबसे ज्यादा 90 to 95% रहता है, बाकी अदर गैसेस होती हैं which is representing the natural gas, right? So we have to depend on how to deal with this energy resource. So if we talk about the energy storage point of view, we can store this energy in two forms, molecules form or the electron forms. But when we talk about the storage in electrical forms, which we are saying nowadays, we are electrical energy, right? But you can look at the electrical energy challenges: poor conversion efficiency, high material cost, and still a large scale storage, right? So we are moving towards the LNG and CNG. LNG also provides some challenges, which also offer some energy, but also provides some challenges like extreme low temperature, minus 161 degrees Celsius is required to cool this natural gas in order to store the natural gas in the form of liquefy. Whereas if we talk about the CNG, we need compressed natural gas to store the natural gas in the form of CNG. It requires around 200 to 250 bar at least. Now you can imagine. Can anyone tell me how much is the car tire pressure? What is 35? What is PSI? Can you convert it into bar? One PSI equal to around 14.6. Oh, sorry, one bar equal to 14.6 PSI, right? So it's almost around 3 to 4 bar. What happens when you inject more air? The car tire burst. It leads to disaster. Now you can imagine that 200 bar or 300 bar, what will happen if it is burst? Still a highly risk associated with the CNG. So we have two challenges with CNG, LNG: low temperature and high pressure. In order to achieve 0 degree, how much energy is required to reach 0 degree in the refrigerator? आपका बिजली का बिल ज्यादा आ जाता ना जब आप ज्यादा रेफ्रिजरेटर यूज करते हो. That means the economic part is also associated. So we have to tackle these two problems. I'm offering, or the researchers offering, a new technology or the promising technology which is known as gas hydrate, where we will deal with this low temperature and the high pressure, both the conditions. So in the layman term, what is gas hydrate? This is water molecule and this is gas molecule. Both of these water and gas molecule comes in contact at suitable temperature and pressure conditions. What are these suitable temperature and pressure conditions? Low temperature and high pressure condition. These water molecules form the cage-like structure with the help of hydrogen bonding, and in these cage-like structure, the gas molecule reside. That's why we call these gas molecule as guest molecule and the water molecule host molecule. Water molecule क्या है? Host molecule है जिसमें आके मेहमान बैठ जाते हैं, वो है guest molecule, right? Clear? So when the 1 meter cube of gas hydrate volume can store 160 to 170 meter cube of gas, Now you can imagine that large amount of gas can be recycled. But when the low temperature and the high pressure condition met. The conditions, the temperature and pressure conditions is varying according to the type of gas, whether it is methane, propane, butane, or any natural gas, CO2, hydrogen, etc. Now the question comes how it looks like. You can see molecular level, we have this hydrogen bonding, we have this methane molecule CH4, and this is the cage-like structure. If we look at the gas hydrate from the naked eye, it looks like ice-like crystal. When you bring this ice-like crystal, the gas hydrate in the environment or the room temperature, what will happen? The temperature starts increasing, the hydrate becomes stable. The hydrate becomes unstable because the temperature for the formation temperature is low temperature, right? So at the environment, the hydrate becomes unstable. What happens? These cage-like water molecule, the cage-like structure starts breaking and the gas comes out from these cages. If you just ignite it, it starts burning. So we also call burning ice. If you look at this map, we have Canada here, we have Russia here, Japan, India, China. All over the world, we have a lot of natural gas hydrate deposits under the seabeds. That means we have a lot of natural gas. I'm not talking about oil. I'm talking about only gas. People are talking about electrical energy, electric bus and something, right? Forget about this something. Just focus on oil and gas. Forget about oil, only gas. Now you can see this much amount of gas is present in the world. If we try to recover or extract this gas from these deposits, we can supply for next 1,000 years. So no need to worry that fossil fuels are going to be exhausted. No. Where these gas hydrate forms or exist in the permafrost region because of the low temperature or oil and gas pipelines or subsea wells, right? जहां पे भी आपका low temperature high pressure condition met हो जाएगा और वहां पे gas hydrate बन जाएगा. I can see that some of you are busy with your phones. I request you to please do not look at your phones. Just look at these slides. It will be more interesting, right? So these gas hydrate structures classified into 3 types of structures: structure 1, structure 2, and structure H, based on their cavities. So or the cages structures. So we have some small cages, medium cages, and large cages. If we talk about the natural gas hydrate deposit in India's scenario, we have 134 trillion cubic feet of natural gas hydrate reserves, which is the 1/3 of the US reserves. If you look at here, Mahanadi Basin, Krishna Godavari Basin, and somewhat in Konkan Basin, which is already discovered. We have this basins where we have natural gas hydrate deposit. But there are some challenges. How you will recover the natural gas? What are these technology? How you will recover the natural gas? Why I'm talking about the natural gas recovery? Because this is the energy resource. So the gas hydrate has several applications like solidified natural gas. I showed you, right? The ice-like structure, naked ice. Another is seawater desalination. For the gas hydrate formation, only water and gas are going to participate. When you will dissociate, you will get natural gas and you will get potable water. So why we are focusing on seawater desalination? We have enormous amount of seawater is available, right? Another is hydrogen storage. Why we only store methane? We can also store the hydrogen gas. Why we are focusing on hydrogen? Because hydrogen gas is cleanest source of energy. But problem with hydrogen storage is this hydrogen is very naughty molecule. So we have to be worried about the hydrogen molecule can go here and there anywhere, right? So we have to monitor the formation condition. Another is CO2 sequestration. I'm not talking about the CO2 storage, I'm talking about the CO2 sequestration. We can store the CO2 for the longer period of time. Where we can store? We can form the hydrate under the seabed or in the depleted reservoir. What are the depleted reservoirs? Where we can already extract the oil. there is no use of these reservoirs, we can store the CO2 for the longer period of time. तो एक हमारा एक प्रॉब्लम सॉल्व हुआ ना कार्बन एमिशन वाला. Now, so the gas hydrate offers energy as well. So we can say that we can supply for next 1,000 years because it is evenly, uniformly distributed around the globe. We have talked about the positive part. Now we have to look at the negative part as well. These gas hydrate, because when you are transporting this natural gas through the pipelines, water comes in contact in the permafrost region. This pipeline, the hydrate formation in the pipeline occurs, and the pipeline, the flow is hindered to flow, and the pipeline burst. In that cases, you can see here, the hydrate looks like this. So in that cases, we have to add some chemicals rather than to prevent this hydrate formation, or we can add some anti-agglomerants by which the agglomeration cannot occur, or some other additives by which the hydrate formation cannot be happen. So in that case, we can transport the natural gas. In case if the hydrate formation occurs, it should not be agglomerate. It should be transported through in the form of slurry, which called hydrate slurry. So the management of these risk hydrate by using some chemical or some other additives, which comes in the part of to study the rheological properties, to understand the flow behavior of these hydrate slurry. This comes under the flow assurance part. I hope it is very clear. So I'm going to show you the natural gas hydrate sample, which is natural sample, which is found in Lake Baikal, Russia, which is the deepest lake in the world, around 1700 meters. And these samples found just below the lake surface of around 300 or 400 meters. It's not working. So we can, oh, what happens? Sorry. No, it's working in this mode. Nice.
वीडियो चलेगी
नहीं नीचे वाला है. Just play this video. I think it's बड़ा करने पे नहीं चल रहा है बेटा. It's visible, right? So you can see I'm holding this natural gas hydrate sample in our hand, and it's extremely safe. Why? Because the cage starts breaking, and I'm feeling the cold water on my palm. And in these cages, the gas comes out and just ignited, it starts burning. So that's why we call burning ice. It's extremely safe. Right? You can see how it is safe. So this natural gas hydrate sample, we can also form this hydrate in the laboratory, and also it's safe, extremely safe. So we have 3 techniques to recover the natural gas from the hydrates. One is depressurization. You can reduce the pressure. Another is thermal stimulation method, in which you are going to increase the temperature. Once you increase the temperature, the hydrate becomes unstable. Another is the chemical inhibitor injection method, in which you are going to add some additives by which the hydrate cannot form and the gas comes out from these cages. Working. So Japan and China has done the pilot field test to recover the natural gas in 2017 and 2019. However, they have not shared the data with the any countries. So Russia is also doing this field test to recover the natural gas, but Russia has a lot of natural gas reserves. India also working on these recovering of natural gas from these KG basin or the Manadi basin, but there are some government policies we cannot go up till once we did not get the approval of government, right? So I just talk about the why this advantage of SNG technology, solidified natural gas, the compact storage, ease of natural gas recovery, stable, extremely safe, and if you look at water and natural gas, we form the hydrate, which is called SNG pellets as well. But there are some challenges. What happens when the water molecules or gas molecule comes in the contact? Do you think that hydrate forms immediately? No. It can form immediately. It can take 1 hour. It can take 2 hours. It's called stochastic process. But there are some challenges. Sometimes natural gas is not going to reside in these cases. So we have some kinetic problems as well the gas consumption. So we have to work on the slow kinetics. Also, we have to work on the rigorous condition of hydrate formation. In order to solve these problems, we have to look at some additives. So for that, we have used some SDS surfactant and other surfactant as well. So before going to start, I will like to introduce some other additives, biopolymers. So we have used some biopolymers, sodium alginate, guar gum, PVA, polyvinyl alcohol, and some other additives, L-xanthan gum. And we have looked at the kinetic data, that how much amount of gas consumption data, how much amount of gas is stored in these cages. And we have observed that PVA shows the highly significant and gives the more gas consumption. You can see here this PVA. And we have used this frozen particles of biopolymers. Right. Please let me know, madam, how much time is remaining. Okay. Now we have used another additive which is called SDS, sodium dodecyl surfactant, and I have used the 0.25 mass fraction, and we have looked at the morphology. Why we are focusing on the morphology? We should understand the hydrate crystals growing in which direction. Because when we are forming this hydrate, when we are storing the natural gas in the form of hydrate, we should understand these crystals behavior. Or when we are extracting the natural gas from these hydrate, we should know the crystals locations so we can recover the natural gas. So we have looked the morphology of the system SDS 0.25, and we have observed in around 10 minutes the complete reactor is filled with gas hydrate. So we can say the rapid hydrate formation occur. But what happens with SDS? This is surfactant, right? Similar to detergent, which can generate some foam. So when we dissociate these hydrate, it releases some foam. But we are not dealing with the foam, we are dealing with the natural gas. So we have to solve this problem because this is the laboratory scale. If we think about the larger or commercial industrial purpose, this large amount of foam, we have to solve this problem. So we have used some anti-foam A agent which can reduce the foam. So we have used different concentration, but here we have used showing 0.125 anti-foam A concentration, and look at the morphology. Here, this is the formation condition when the hydrate form. We have same morphological, same behavior observed. In the 10 minutes, almost the hydrate is the reactor is fully filled by hydrate. Now I'm focusing on the dissociation part. At the time scale of 60 minutes, when the temperature is room temperature, there will be no hydrate particles, no hydrate crystal. That means whatever inside in the reactor, is the completely foam. And we have waited till 8 hours, still the foam is there. This is only for the reactor, small scale. think about the large scale. So we have introduced the antiform A and 0.125 concentration. We have used different concentration, but 0.125 gives the better optimal concentration, and we have observed at 60 minutes and 2 hours, which is almost half of the foam is reduced, and if we waited for another 2 hours, there is no foam. So we can say that we can use the SDS. Along with this, we can store the natural gas in the form of more gas consumption, and there is no problem. Now moving towards the another additives, which is called as THF, tetrahydrofuran, which behaves as thermodynamic as well as the kinetic promoter. So if we look at this figure, we have this phase equilibrium curve, and we have this red symbol is showing only with methane and water. And this zone is known as hydrate zone. This is non-hydrate zone. And when we introduce some THF, other additives, and we observe the shifting of hydrate phase equilibrium curve towards the right, which shows significant behavior of the promoting the hydrate formation. That means if we use the THF, the hydrate forms quickly and more kinetics as well as thermodynamic behavior. So if we look at this, I'm going to play this small video. So you can see in this, this is aqueous phase, water phase, this is gaseous phase. A thin layer of hydrate forms, and after around 3 minutes or more than 3 minutes, there is no further diffusion of gas. There is no further hydrate formation, right? But and the condition is methane and water only, and temperature is 1 degree Celsius and 9.5 megapascal. Is this 1 degree Celsius better than minus 161 degree Celsius? Definitely. Is this pressure 9.5 megapascal is better than 200 bar, 20 megapascal? Definitely. But still, we can see that there is no further hydrate formation. So we have introduced the THF, which is around 5.5 mole%, and we have reduced the pressure 9.5 to 7.2 and increased the temperature. And we have looked at the behavior of the hydrate formation. Now here you can see, we call the rapid hydrate formation. So quickly the hydrate crystals are forming within the fraction of seconds, and it's completely filled this glass vial. This represents the rapid hydrate formation, and this also shows the THF can be used as thermodynamic promoter as well as the kinetic promoter. Now we are moving toward the 10 degree. We can also form the hydrate at room temperature. So why from this we can say that this technology economically viable as well. Extremely safe, economically viable. So we have used the THF in non-saline system and saline system at, I showed you around 7.2 megapascal. Now I'm going to show you at 5 megapascal, that is 50 bar, and 3 megapascal, 30 bar. The temperature is 10 degree and concentration is 5.5 mole%. So we have looked at the morphology. In 10 minutes, the hydrate forms entire in the aqueous phase towards the water phase, and there is no further formation of hydrate. When we deal with the, I also look at how changes the crystals are forming. So we have used the food color dye, which is red color, and look at the morphology. In non-saline system, I also look at the 3 megapascal, similar behavior. But when we look at the 3 megapascal saline system is 3% of NaCl we have used, and we have observed the sluggish hydrate formation. That means the hydrate cages or hydrates are not that much thicker, not strong. So we have looked at the gas consumption kinetic data, and we observed there at 5 megapascal almost in saline system, non-saline system, the gas consumption is similar. However, in 3 megapascal in saline system, the gas consumption is not that much. So we have to think for another, we have to find out the another additive which can also provide the same gas consumption at 3 megapascal for the saline system, because we have a lot of seawater is available. But we also look at the dissociation morphology, and we have observed in the saline system, the hydrate dissociates faster. Why in 5 megapascal and 3 megapascal? We can see that there is a difference of 9 minutes exactly. So because of the in saline system, the hydrate bonds, the hydrate crystals are not so much strong, not thicker. That's why they dissociate so fast. Now I'm coming back to the first slide. So this we have talked about the energy storage as well as the energy recovery. Now I'm coming back to the decarbonation, sustainable future. How we can store this CO2 in the form of hydrate. We have a lot of methane hydrate around the globe. If we inject the CO2 gas, which is already captured, we can inject the CO2 gas. These CO2 molecules, bigger in size, can push the methane molecule and reside over there, and we can get this methane at the surface. So we can get energy recovery, and we can also get this CO2 sequestration in the form of hydrates. So this is showing the mixture of CO2 and N2 we have in places, we recovering this CH4 and we form the CO2 hydrates. If we talk about this technology, the technology TRL level technology readiness where we have achieved, so we have achieved the TRL level 7. We have already demonstrated this level CO2 hydrates technology in Japan, China, Russia, and Singapore. In India also we have done this at the laboratory scale, but still waiting for the another TRL level. These are several approaches how to store the CO, how to achieve the CO2 sequestration. 3.5 meter cube of gas hydrate can store 1 ton of CO2. These are approaches for capture and separation of CO2. So I'm here only focused about the hydrate based CO2 separation. So if we thought about the CO2 sequestration in the deep under the seabed, if we look at here at 349 meter, the temperature 8 degree, we are going deeper, temperature is reducing at 1.6. So we can also achieve the hydrate formation, CO2 hydrate formation in the deep lake. But there are some several challenges like hydrate instability at the sea floor, low hydrate conversion. Another type dissolution of CO2 hydrate under the seawater. It can release some seawater current and it also affect the aquatic life, which is not good. So we have to focus on these issues while sequestering the CO2 under this. When we're talking about the liquid CO2 and the CO2 hydrates. So we have this system here, reactor, where we are forming the CO2 hydrates. We have CO2 gas. We are forming the CO2 hydrate. We are mimicking the real situations for the temperature pressure condition, and we observe the low with the comparison of saline water. We have observed low slow kinetics when we use the sea water. So we have to again identify some additives. So researchers already working on these additives like amino acid, right? So still we are focusing on these which additive which is good for the environment and provides the better kinetics. So I'm moving towards the conclusion slide, right? So we can say that a great opportunity to utilize the seawater for SNG technology, gas hydrate potential source of energy, CO2 sequestration, hydrogen storage. Again, we can also use this technology for the clean source of energy. We can store the hydrogen in the form of hydrates. If we talk about the energy storage point of, we can use the SDS for more gas consumption, and we use the antifoam A along with the optimal. We can achieve, we can tackle this foam problem, and we have also looked at the morphological properties to understanding the designing of the reactors and the flow assurance problems. So I have published several papers on the hydrate technologies. So I would like to acknowledge my students, those who are working at Earth Cryosphere Institute, Russia. So these are some of my master student, PhD student, along with some of my colleagues. Yeah. So if you have any question, feel free to ask right now. Session is open. If you want to chit chat with me, you can ask question outside the this hall, or you can take my email ID. Yeah. And thank you for listening my talk. एनी क्वेश्चन? इससे ज्यादा मैंने कभी लेमन लैंग्वेज में अपने कभी भी टॉक दी नहीं है. दैट्स व्हाई आई आस्क व्हिच बैकग्राउंड यू हैव. राइट? सिंपल है ना, वाटर मॉलिक्यूल गैस मॉलिक्यूल पास में ले आओ, मिल जाएंगे, गैस हाइड्रेट बन जाएगा. Not in open environment, it's in a closed environment, right? In a reactor. Any question? आप लोग सब मोबाइल में ही बिजी थे, क्वेश्चन क्या पूछोगे?
ऐसा लग रहा है कि सब कुछ समझ में आ गया, या तो सब कुछ समझ में आ गया या तो सब कुछ ऊपर से गया. Let's hope it's the first thing. So, thank you, Dr. Gaurav. So we are having collaboration with them. So questions अभी पूछ लो, रशिया जाने का मौका मिलेगा. He is having collaboration. We too are having collaboration. रशिया, मॉस्को. अभी थोड़ा रिस्की है, but they are doing quite good research. अभी सुबह में जो सर बैठे थे उन्होंने भी बोला, they are doing quite good research in terms of nuclear power. Russia is one of the longest standing ally of India also. Like we are good friends. We are also good friends. तो जिनको अगर नेचुरल गैस हाइड्रेट में रिसर्च करना है वो बाद में आके भी मिल सकता है. एंड सो यू कैन बी इन टच विद सर. सो थैंक यू सर.
मैं थोड़ा सा करेक्ट करूंगा, रशिया में कोई प्रॉब्लम नहीं है, प्रॉब्लम यूक्रेन में, तो यूक्रेन मत जाना.
नो पॉलिटिक्स.
Okay. So thank you for inviting me, Professor Sanket Joshi, here and giving this wonderful opportunity to meet the students. Thank you.
Okay, so now we will move on to the oral presentation part. So Moses Paul from VIT. Design and application of ohmic heating to study the effect on tomato puree. अपनी presentation दीजिए, मतलब आप उसमें लगा के ओपन कर दो.
ये हटा दूं क्या सर?
ये क्या है लॉजिक?
मुझे पता नहीं.
इधर पे होगा ना, इधर नहीं है. You have 5 minutes, okay? I think everything simple. Easy.
It's not something.
I'll just move this slide. Please start. Let's start from there. Start from there. Okay, your time is not Good evening, everyone. So this is Joy Duncan, and I'm from VIT Vellore, studying 4th year chemical engineering. This is my teammate. This is Moses Paul. We are from VIT BTech Chemical Engineering, 4th year.
Hi guys, and this is Industry here from VIT Vellore.
So today our topic is design and application of ohmic heating to study the effect of tomato puree. So as we start this topic, let me ask you one question. Does anyone know what Ohm's law is? We studied this in 9th and 10th. Ohm's law. What's the formula for that? V is equal to I. Yeah, right. So what does it say? Voltage is directly proportional to current and resistance, right? So that's what we are basically giving you a gist of it. We'll go and break down the main topic and help you understand the topic. Thank you. All right. So why tomato? Why are we chosen tomato for this research purpose? So basically, the global waste program that is conducted in 2012, 16% of the annual tomato production is discarded, either from the production loss or through consumer loss. 16% of it is discarded, and it has a loss in economic value and environmental burden. So this, in turn, makes us come into a solution that makes us turn waste into value-added products like puree and lycopene extract. So what is value-added products from tomato? So tomato purees and juice that we already know, and lycopene extract. So this is lycopene is an pigment that gives red colored tomato, and it also acts as an antioxidant that reduces risk of cancer and acts as a heart health, supports heart health. So what are the characteristics of tomatoes that is appropriate for ohmic heating? So one is it is highly electrically conductive, and the mechanism is it helps in uniform volumetric heat production, unlike conventional methods. And cellular structure. Here, ohmic heating helps in protecting and preserve the quality and texture of tomato. And why ohmic heating especially? Because the use of
All three of you will be speaking.
Yes.
Audience, kindly listen to what the speakers are offering you. Okay, so please do not make any kind of noise in between. That is disturbing everyone. So please maintain the decorum of the conference. Otherwise, you can leave. There is no compulsion. Okay? Thank you.
So application of ohmic heating. So what are the uses of application and in ohmic heating? That is, can be used in rehydration, pasteurization, sterilization, fermentation, and particularly effective in viscous and particulate foods like tomato puree. Benefits, yeah, there are many benefits for this. That is efficiency wise and quality retention wise and extended shelf life. As in, if suppose a tomato puree can stay for 2 months, by ohmic heating, it can be extended to maybe 3 months or extra 2 months more. The research objective of what we have taken is aim, design and fabricate a lab scale ohmic heating unit to process tomato puree. And the key area studies is the effect of voltage gradient, conductivity, time temperature, and impact of lycopene in tomato puree. And that will be guided by my teammates.
So next, the process is like how do you prepare the sample? So first we got all the mandatory items that all the stuff, and then the size of tomatoes is same of size and color, because we need proper weight to take the proper results. So the first process is putting it in a boiling hot water to blanch the skin of a vegetable, and then the seeds are being removed, because if the seeds are present, then the bacterial growth is more rapid and faster. So the seeds have been removed, and then using the wet sieve like analysis method, we have got the puree of tomato into stuff, and then the particle size is being seen as 0.1204 meter using wet sieve like analysis. And this particle size plays a crucial role in the puree juices, because the what is the process of particle sizes? Particle size mainly says the bacterial growth, rheological, and then the flow of the puree. So next also we followed by all the process, we have done the streak like analysis which say the bacterial growth of the puree. Next my teammate will be continuing.
Yeah, the main theme is like ohmic heating, the fabrication and design of the ohmic heating. So the unit is designed with the equipments of the chamber, which is 8 mm acrylic sheet, and it is a dimension of 10 into 3 into 5, which is height, the breadth and the length. And the electrodes, which is the stainless steel for conducting of the electricity. And the power source is a voltage meter, for the difference in the voltage between the gradient is 10 to 140. And the measuring tools we are using is the voltage digital multimeter, that is the voltage, current, resistance, and the portable pH meter is used for measuring the pH And this is the process like the unit which we're having in our college. And this is the experimental setup. For the voltage gradient we are using is 10 to 15, 20, 25 and 30 volt per centimeter. And we have taken 3 time intervals for each sample we are doing. And the measurement and analysis which the results we have done is and discussions is the lycopene content, the temperature and the heat conductivity and the energy consumption, the pH and the efficiency. We'll just look to the results and discussions.
So now and then we are going to see about the first results, which is like conductivity. So first thing what we saw after we got the results was shown in the graph. As the ions get into some place, the conductivity of the material got increased. So it's shown in the graph. And then when you come to as we increase the voltage, this thing also started to increase, and then also we have used the formulas like AT square plus BT plus C, and then we used to take this, and then ABC are the constant values in it, and the further results will be continued by my teammate.
So we have taken a range of temperatures ranging from 10 to 30 volt per centimeter in 5 degrees Celsius basis. So I'll run through the main points quickly. Temperature rises linearly from 25 to 96 degrees Celsius. 96 degrees is the maximum temperature that we can take from Curie. Higher voltage gradient results in faster heating due to increased resistance generating. That is because V is equal to IR, so higher voltage obviously equals higher resistance. And the pH study done here is with decrease in pH decreases with increasing voltage gradient. That is, we can see from here in the graph that is given here.
Yeah, the energy consumption and the lycopene content. The energy consumption has the increase in the temperature and the voltage gradient. Usually the energy consumption will be more as the increase in the voltage, increasing the current actually. And it is observed with this value of specific performance coefficient and the lycopene content, which is a very important thing as a nutrient in the food. And it is degraded in the conventional method, which we use as in a traditional way. And this in contrast, the ohmic heating, which can compromise it as a reduction in the degradation of lycopene content. And this is a graph. And we are into final conclusion. The ohmic heating, this is the conventional method which can compromise the traditional way of heating. And the main thing is we are into the electropolarization effect on microbial load required for the further study. We are into that, and we are thankful for our college, which they funded us for designing the unit. And we are thankful for this opportunity for us to give a wonderful chance to present in this. And thank you for the Amity University.
I
Are
have
there any questions from the?
Yeah, I just want to ask, have you studied only the lycopene, or there are other contents also which you study? Because you have shown only lycopene. So have you seen any other nutritional content just to see whether it is getting, maintained or?
Scope here is to examine tomato puree and observe how ohmic heating is affecting the properties and textures of the ohmic heating for extended shelf life. So for now, we have studied only lycopene because that lycopene pH and everything comes under lycopene itself because it's directly affecting the lycopene extraction there.
Because why I'm asking this is in tomato, is it only lycopene is not giving the nutritional value, right? So other things, studying them also plays an important role if you want to see the nutritional value of the puree, right?
Yeah, ma'am, it's actually that, but only here we studied only about this content, lycopene content. So we also we mainly focused on like other stuff, how it increases, that temperature increases.
Okay.
Question from my end. What is actually the role of lycopene content in human body? How will it help you?
It is an antioxidant and it helps in various health issues like preventing from the health issues like the bones and like cancer kind of.
Also, even in some kind of heart issues, also it's helpful, ma'am, to resolve it.
Okay, actually, prostate cancer, one of the best methods to prevent prostate cancer, that is lycopene as a nutraceutical. And that could be the reason why you started off with lycopene. Okay,
Okay.
thank you.
Thank you, ma'am. Thank you, guys.
Is Gargi here? Participants, you have strictly 5 minutes to present your work and there will be 2 minutes question and answer session. So please stick to the time.
Good afternoon, everyone. My name is Gargi. I am research scholar in the Department of Bioscience and Biotechnology, Banastili Vidyapeeth. And I'm going to present some research work of my PhD. And the topic for the presentation would be arsenic-induced morphological changes in Vigna mongo varieties and the mitigating effects of biaxial iron nanoparticles. And let me start from the introduction part. Many type of environmental stresses are increasing in the environment, and these stresses adversely affect the crops. And mainly abiotic stresses such as cold, salt, and heavy metals affect crop production. And soil contamination by heavy metals and metalloids is a serious concern and a big challenge for the food security. Arsenic belong to the semi-metallic group, and it's also known as the king of poisons. And it is the first toxic element that causes serious effects in human population and also the plants. And the sources of arsenic may be geogenic, biogenic, and anthropogenic sources. And the affected states in India, these are the brown part in the map shown, are the states that are affected by the arsenic. And in plants, the arsenic can affect the leaf, stem, and roots in various forms, like causing diseases and decreasing number of nodules or cellulose depositions, and many others. And I have chosen the crop Vigna mongo because India produces many pulse crops. One of them is Vigna mongo, that is black gram. It is rich in many compounds like protein, carbohydrate, fats, and have many health benefits like heart healthy. It keeps the heart healthy, improve digestion, promote hair growth, supports bone health, and many others. And it also a rich source of protein. And the producer states of black gram would be the Uttar Pradesh, Andhra Pradesh, Maharashtra, Tamil Nadu, Madhya Pradesh, Rajasthan, and others. And also the India is a 70% the 70% production of black gram comes from the India. And also the larger producer and consumer of black gram is India. And the nanoparticles, that we know that the particle size less than 100 nanometer are the nanoparticles, and which shows some peculiar properties over the bulk material, and it can be synthesized various modes like chemical, physical, or biological mode. And the beneficial and harmful properties depend upon its size, shape, composition, and concentration. And the plant from which I have synthesized the nanoparticle is Cymbovogon citratus, that we known as commonly known as lemongrass, and it is a medicinal plant and used in many forms. It can be used in teas, and also the lemongrass oil is used, and it also rich in very compounds, that is phenols and flavonoids, that is a good source for generating or creating nanoparticles. Then iron is an essential micronutrient for plants and plays a critical role in the following metabolic process, like chlorophyll formation, electron transport in photosynthesis, activate enzymes, and DNA synthesis. And iron has the potential to elevate metal toxicity by limiting the metal uptake in different plants. And I have chosen the biosynthesis mode because it has many advantages, like it is non-toxic, cost-effective, and eco-friendly, and many other. And my work has been divided in 3 portions. First one is the screening of Vignamogo on the different concentrations of arsenic, and 5 varieties were chosen. And after 10 days of sowing, different parameters of morphology were taken, like the root length, shoot length, their fresh weight, dry weight, leaf area, and other. And the second one is the biosynthesis and characterization of iron metal, and leaf extract of lemongrass were taken, and with the metal salt in 1:2 ratio. And then characterization was done with the
Gargi, please come to the conclusion. Experimental layout, leave it.
Here, the germination percentage of the 5 varieties that I was chosen shows the different varieties, and then the growth parameters that I studied for the 5 varieties. And on the basis of these parameters, it can be concluded that the one variety that is PU1, and the second one PU31, one is tolerant, that is PU1, and the sensitive one is PU31 for the arsenic. And next step was done, that is characterization of magnetite nanoparticle with the UV spectrophotometer. And then FTIR, different bands were shown, and 690 band shows the presence of FeO that confirm the formation of iron nanoparticles. And then the particle stability and size formation was also calculated by the zeta sizer and potential. And then XRD was used for the crystalline size determination. And then SEM was SEM provided the image that was cubic shape nanoparticles were formed, and the EDX data given that the Fe and oxygen compounds were also present. And then after synthesizing metal nanoparticles, these were given to the 2 varieties, that is PU1 and 31, in different concentration by foliar application. And by examining the morphological parameters, I can conclude that the 50 mg per liter nanoparticle were the most effective one in PU1, and also in the PU31 variety that was sensitive to the arsenic. And I can conclude in the last that iron nanoparticle can be used for the protection of can be used to protect the plants from heavy metal stress and increase the resistance to arsenic stress. And further study of biochemical and molecular parameters can help how the arsenic was mitigated by the iron nanoparticles. Thank you. And I would like to thank my guide, Professor Nilima Kumari and Vinay Sharma sir, and also Professor Ina Sastry ma'am. Thank you.
Gargi, did you use any method of synthesis in which you used a biological method for synthesis of nanoparticles?
Ma'am, I use the biological source only for synthesizing iron nanoparticles.
Which plant you used?
Sembobogon citratus, that is lemongrass.
Okay, thank you.
Thank you, ma'am.
And Anju Sogarwal.
Good evening, everyone. I am Anju Sogarwal, research scholar in the Department of Bioscience and Biotechnology, Banastali Vidyapeeth. I am here to present my research work on the silicon's protective role in wheat, alleviating fluoride toxicity and enhancing suberin and cell wall strength via cellulose synthase A4 gene expression in wheat cultivar. As we know, the fluoride is the most electronegative and highly reactive element, and it is the anion of fluorine, and it is liberated into the environment through the volcanic emission and rock weathering. The liberated fluoride become mobile and enters the soil, groundwater, crop, fruits, and vegetable. As the sessile organism, plants are sensitive to fluoride. Fluoride cannot be fully detoxified by plant and posing risk to both plant and animal health. This image shows how fluoride induced toxicity in plant. The fluoride enters the plant by the root and it transported to the shoot and leaf by apoplastic and symplastic pathway through the xylem and phloem. Once the fluoride enters in the cell, it generates ROS and causes toxicity, which ultimately reduce the plant growth and decrease the crop yield and lower the grain quality. Wheat is a global important crop,
Anju,
highly sensitive to
Anju, please come directly to your work. Introduction part you can skip.
silicon mineral nutrition in plant is important for maintaining healthy development and increasing the stress resistance. Silicon is the one of plant nutrient providing significant advantage for growth and yield of the crop plants. This slide shows the experimental setup. These are the experiments were used to analyze the evaluate effect of elevating silicon in wheat cultivars under the fluoride stress. The effect of silicon treatment on MDA and S2O2 content under the fluoride stress. This image shows the MDA and S2O2 content were increased in the fluoride stress plant, and the rise in the MDA and S2O2 content indicate the fluoride-induced oxidative stress. However, the soil application of silicon decrease the S2O2 and MDA content, and which shows the silicon phytoprotective role in wheat plant. Effect of silicon treatment on activity and antioxidant enzyme under the fluoride stress. The enzymatic antioxidant have been considered the first line of defense to respond to stress. We examine the three enzymes, catalase, APX, and SOD. All enzymatic activities were decreased in the fluoride stress plant. However, the silicon application further enhanced the enzyme activity, which shows the improvement the antioxidant enzyme and decrease the ROS generation. The effect of silicon treatment on cellulose synthase A4 gene expression under the fluoride stress. The cellulose synthase gene were decreased in the fluoride stress plant, and as we can see the image. However, the silicon enhanced the cellulose synthase A4 gene expression. Effect of silicon treatment on suberin component under the fluoride stress. The suberin is the polymer and it is made up of many monomers. The fluoride, the fatty acids, omega hydroxy fatty acids, secondary hydroxy containing fatty acid, omega dicarboxylic acid fatty alcohols are the major component. Fluoride decrease the all major component of suberin, and however, the application of silicon enhance the all major component of suberin, as we can see the image. In conclusion, the fluoride stress increase the ROS production, enhance the oxidative damage, increase the antioxidant enzyme activity, and reduce the suberin deposition and down-regulate the cellulose synthase A4 gene. While the silicon application reduce the ROS production, decrease the oxidative damage, increase the antioxidant enzymes, and enhance the suberin deposition, and upregulate the cellulose synthase gene. And this image shows the silicon improves the apoplastic barrier and indicates by the increasing suberin deposition. Silicon can be an alternative to conventional chemical fertilizer. Chemical fertilizers are often harmful to the consumers, farmer, and environment, and for which consumers are increasingly seeking safer alternatives. It is non-pollutive, non-corrosive, non-toxic, and it is naturally present in all the plants. These are my publications. I'm thankful to Vanastali Vidyapith for providing research facilities, and I am also thankful to my supervisor for her guidance, and I am also thankful to CSIR for providing financial support. Thank you.
Wait, Anju, I just want to ask a question that have you done this only for the fluoride toxicity, or it can alleviate the toxicity of other metals or other substances as well?
Only fluoride. Actually, silicon. I used only silicon.
But is there any report?
Fluoride toxicity.
But is there any report available for other kind
Yes,
of?
ma'am.
What are those? Like other than
Others, ma'am, silicon dose 200 mg per kg and 300 mg per kg.
नहीं आई एम आस्किंग कि आप सिलिकॉन जब यूज करते हो उससे यू आर एलिविएटिंग द फ्लोराइड टॉक्सिसिटी बट इज देयर एनी अदर सब्सटेंस अदर देन दिस फॉर विच इट हैज बीन रिपोर्टेड
I don't know. No idea.
ओके
Mishra, Sonal Mishra.
Good evening, all of you. Myself, Sonal Mishra, Research Scholar, Department of Bioscience and Biotechnology, Banasthali Vidyapeeth, under the supervision of Professor Nilima Kumari. Here I am present my research work on the fluoride-induced changes in morphological, biochemical, and physiological parameters in oats cultivar. Introduction: as the demand of the as the population increases, so the demand of crop also increases, but there are a lot of environmental factors which reduce the crop to grow, and there are major factor in which the abiotic stress. I am focusing on the fluoride stress, which is highly present in the 0.3 gram per kg is found in the earth crust, and it is one of the nature 13 most prevalent element. There are the source of fluoride. There are major two type of source: natural and anthropogenic sources, in which the natural sources, mineral source, and atmospheric sources. In atmospheric sources, fire forest fire and volcanoes are produce fluoride into the environment. And anthropogenic sources, there are the man-made or industries which incorporated the fluoride, or there are many type of stresses in the environment. Here we can see the graph. There are in India 200 districts across and 22 states which are highly affected by the fluoride stress. And there are major states which is in which the Gujarat, Rajasthan, and Punjab, which is worst affected from the fluoride stress. Now I am focusing on the oats plant. As we know that the nowadays we highly use the oat as a human. Highly use the oat cultivars. Around the 27 million hectare and 40 million tons of the oats are produced worldwide. And in India, there are UP, Bihar, and Haryana, Odisha, and West Bengal. There are some states which produce the oat cultivars. Oats is the versatile crop that can be produced in their natural habitat, which include desert or infertile regions with chilly climates, and is the major. It is abundant in antioxidant, vitamins, mineral, and starch, proteins, and especially beta-glucan, which is helpful for the digestive system. Here we can see that the oats plant affected by the fluoride stress in different concentration, like 100 to 500 mg per kg. It reduced the growth of the oat cultivar when the concentration of fluoride was increases. Here the growth condition which was selected for the plants under the growth chamber, and there are 4 different concentration of fluoride was selected in which the oat cultivars was grown. 6 oat cultivars of screened in response to fluoride stress. The screening was done on morphological parameter. On the basis of morphological parameters, there are 2 cultivars was selected. One is tolerant, which is JHO 99-1, and one is the sensitive oat cultivar, which is the JHO 822. Here are the material methods for which performed.
Now come on the results. Effect of the fluoride toxicity on morphological parameters. As I earlier told, under fluoride states, all 6 cultivars show the overall reduction in the growth of plant in comparison to the control. As shown in the table, there is the JHO 99-1, which shows the minimum reduction as the concentration of fluoride increases. But JHO 822, which show the maximum reduction in the shootland, rootland, fresh weight, or
Sonal, please wind up as early as possible.
here the fluoride by accumulation was checked, in which the fluoride accumulate higher in the root in comparison to the shoot in both the variety. But in the tolerant variety, there is the less accumulation of fluoride in comparison to the sensitive variety. I was also checked the chlorophyll content. As the concentration of fluoride increases like 200 mg or 400 mg per kg, the concentration of chlorophyll was decreases. And the effect of fluoride on H2O2 and MDA content, as the ROS increases due to the fluoride stress in the plant, so the H2O2 and MDA content was also increased. And now plant elevate the ROS or fluoride stress. It induce the flavonoid or polyphenol, which is the non-enzymatic antioxidant, to elevate the fluoride stress. So here we can see that the polyphenol and flavonoid concentration was increased as the concentration of fluoride rise. And same as the pattern follow on antioxidant enzyme like SOT catalase and APX. And major effect of fluoride on chlorophyll fluorescence was measured on the basis of the ETR and yield values. Here the in control, the higher yield comparison to the 200 or 400 mg per kg of fluoride stress. Now, summary and conclusion, I concluded that the if fluoride stress was given to the cultivar, which reduce the growth parameters and increase antioxidant activities to cope up the fluoride stress or other stresses, and reduce the photosynthesis, which directly related to the yield, reduce the yield of the crop. And I suggested that the variety, which is JHO 99-1, which we can use for majorly affected area of fluoride, because it is tolerant to the fluoride stress compared to the all 6 varieties. And I am thankful for my supervisor, Professor Nilima Kumari, and Professor Vinay Sharma, and Professor Ina Adit Sastri, Banastali Vidyapith. This is my publication. Thank you. जी.
Why did you select only fluoride? Like, what is the reason for selecting fluoride as a?
In Tonk area, there is in soil there are major affected area, which मतलब जो राजस्थान में specific Tonk area जो हम यहाँ पे study करते हैं, वहाँ पे highly fluoride ही present है soil में. So हम वहाँ पे कैसे grow कर सकते हैं different type crops को, और कैसे उसको fluoride को हम elevate कर सकते हैं different methods से.
Okay.
Thank you.
Thank you.
Next is Gaurav Rawat. Gaurav.
So, good afternoon to one and all present here. My name is Gaurav Rawat. I'm currently pursuing my master's in industrial microbiology from Amity Institute of Microbial Technology, Amity University, Rajasthan. So, today I'm here to talk about a topic that not only embodies innovation, but also it addresses the pressing issue of the times, which is the sustainable biosurfactant production using waste frying oil. So, looking on to the background part, with the increasing need for sustainable and eco-friendly alternatives, the development of biosurfactant offers a promising solution to the environmental and health risks which is posed by synthetic surfactants. Biosurfactants are natural biodegradable, produced by microorganism offering environmental benefits over synthetic surfactants. They possess excellent functional potential, making them suitable for diverse application ranging from crop cultivation in terms of agriculture to crop storage, and from personal care products to industrial grade products. Now, the sophorophyllipid part. A sophorophyllipid is a type of biosurfactant which offers versatile applications due to their antimicrobial properties and in effectiveness in cleaning. They are environmental friendly, biodegradable, and have potential in industry like food preservation and cleaning. Moving on to the waste frying oil part, which is the waste part of many restaurant or food chains. Using waste frying oil as a feedstock for sophorophyllipid production repurposes a common waste product, reducing environmental pollution and supporting the circular economy model, which converts waste into valuable resources. So the research aims to develop optimized production methods for sophorophyllipid production using waste frying oil, focusing on maximizing yeast and yield and purity. It also seeks the access and efficacy of these sophorophyllipid in environmental applications. Significance and impact. The research addresses critical environmental concerns by repurposing a waste frying oil into valuable sophorophyllipid and its industrial applications. So the methods used are microbial selection, which in my case was Candida bombicola, a common yeast found. Then the media designing or the media preparation, which in the case is glucose yeast peptone agar. Then the microbial cultivation, which is the isolation of microbes, and then
Please,
contamination with the common
excuse
part.
me, please go to the results. Go to the
Okay.
results.
So these are the method which I've used: ODPH, E24 index, and the oil spreading methods. These are the results. I have tried the results with two parts. First with as a biodetergent. This is the before treatment. The first one is the dirt part, which is you can see the after part. It is effective in cleaning the dirt. Then the oil spills, which are also being effectively cleaned by the sophorophyllipid. Then comes the fruits treatment with the sophorophyllipid. It is not quite significant in treating the bananas, but you can see the difference in the tomato, which is this one is a control part in before treatment, and this is clearly visible that it shows a prominent result in preserving or increasing shelf life of the tomato. In the discussion and results part, I would like to say that the waste frying oil is successfully converted into biosurfactant, demonstrating a cost-effective and sustainability. Environmental benefits are also showcased, the reduction in environmental impact that compared to traditional surfactant produced. Product applications highlighted the potential use in food preservation, biodetergent formulation, agriculture, and emphasizing versatility. Contributed to a circular economy by utilizing waste product and promoting eco-friendly industrial practices. The future direction for this research is focus on optimizing large-scale production, testing biosurfactant in personal care products, and exploring the role in remediation for sustainable agriculture. At the last, I would like to express my deepest gratitude to my guide, Dr. Sanket Joshi, who is seated here, for his invaluable guidance and encouragement, insightful advice throughout my research journey. My sincere thanks to Dr. G.K. Asiri, Director AIMT, for his unwavering support and leadership. I am also grateful for AIMT for providing this resources and environment that enabled to carry out the research successfully. Any question and answers would be entertained. Thank you.
Gaurav, in 1 minute, please explain how you produce the biosurfactant. Have you produced the biosurfactant from the waste frying oil?
Okay, so yeast that is Candida bombicola is taken and it is grown or cultivated in a suitable media, which is in my case glucose yeast peptone agar. Then it is then tested for its efficacy in biosurfactant production using trial phases in batches. So in batches, I have taken a seed culture, which is a pure culture with 2% of waste frying oil, and rest the glucose yeast peptone, that is a culture media. Then it is then cultivated in batches, which after in 48 hours, and then it produces the biosurfactant. which is then extracted and screened through various analytical methods. And then the application, which is then done on clothes as a biodetergent and as a biopreservative.
Any time difference you can mention in the production process?
No, the more you cultivate the microbe, the more it will produce the biosurfactant. Like the 24-hour batch, it has produced a biosurfactant, but it is not that concentrated. But when you go through the cultivation phase of 48 to 120 hours, it produces a significant amount of biosurfactant.
So the time is an important
An
important factor.
Yeah.
Okay, thank you.
Thank you so much, ma'am.
Next is Harshita.
So good afternoon, everyone.
Harshita, please keep it short.
Okay, ma'am. Okay, ma'am. Okay, ma'am. So my topic is rhamnolipid production and their application in soil bioremediation and agriculture. So we start from rhamnolipid. We call it nature's own warriors or for the soil and crop health. So I have a question first. So guys, anyone ever thought that soil has the solution of cleaning up the soil and making the soil more fertile or can increase the agriculture productivity? Yes, it has. So if we talk about biosurfactant first, so they are nature's they are the surfactant which is produced by nature. We call it, if we talk about rhamnolipid, so rhamnolipid is mainly produced by Pseudomonas, and it has some special kind of qualities or the abilities, like they are produced naturally, they are biodegradable, and they can reduce the surface tension. And now, if rhamnolipid, I have divided rhamnolipid is divided into two parts. First, they are warriors. Second, they are boosting the agriculture. How? Because they have the ability to dissolve the toxic pollutants, and secondly, they can improve the plant growth. This is the process. So normally, if I say I have selected the samples from different soil areas. First is garden soil, then petrol pump soil, and diesel contaminated soil. Then we will move to the Pseudomonas isolation. So Pseudomonas isolation was done by using the special media, the Pseudomonas agar-based media and the nutrient agar. Second, if we talk about the identification, so for the identification, we have used the biochemical assays like catalase, oxidase, indole, and extra. And if we talk about biosurfactant production, so the production was done in the two form. We divided it into the three parts: first batch, second batch, and third batch. And all the batch shows equally the same growth or some kind of different. Second is the screening. For screening, we have done the oil spreading assays and E24 emulsification index. These are the analytical methods I have used. First is oil spreading, emulsification index, pH measurement, and optical density. These are the results. Firstly, I have isolated the Pseudomonas. So out of 20, 6 to 8 strains were identified, which was similar to the morphology of Pseudomonas. Secondly, biosurfactant production. So it was done in 3 batches. First batch has moderate production. Second batch has the same, but the third batch has the highest production. If we talk about emulsification index, and so all the 3 batches has emulsification index, but the second batch was normal, which is 50% with a 50% index, and the third batch has 55% emulsification index. Third, if we talk about the oil spreading, so here this is a video of oil spreading, how clearly that is making the zones. And zones, it's almost around 2.8 centimeter. And now, for applications, I was worked on 2 applications. First is oil spilling, and second one is the soil bioremediation. And this is the most interesting one. What I did was I have selected the soil sample. I have divided it into the 3 triplets. The first one is the normal soil. The second one is the soil which is treated with oil, which was contaminated. And the third one is showing the soil which was contaminated and then treated with biosurfactant. In the first one, you can clearly see the growth, which is
a normal soil, out of 30, 12 to 15 plants were grown. In the second one, only one plant were grown. And if we talk about the biosurfactant one, out of 30, 26 plants were grown, and you can clearly see the impact of biosurfactant. And here is the discussion why I have used the waste frying oil, because they are cost effective and they have some sustainability. And here we are talking about the waste management also. And if we talk about future directions, so yes, we can optimize production conditions like aeration or pH. And biosurfactant is just not about agriculture or increasing productivity. It is in the industries like drug delivery, cosmetics, and industrial cleaning. And here I would like to thank Mr. Sanket Joshi. I have completed my dissertation under his guidance, and all the faculties from the department for their guidance and support. Thank you.
Harshita, you said that you got the highest production in the third batch, right? What is the difference between the second
Ma'am,
batch and
as
third
I
batch?
have selected different soil samples. So the strains I have got from them, I divided into batches. First batch was garden soil. Second one has the strains that were identified from the petrol pump soil. And the third one has diesel contaminated soil, which has which produced highest pseudomonas. And for the production of biosurfactant, the production is highest in the third batch, which has diesel contaminated soil.
So you should actually say that the one which was contaminated with diesel gave you more production rather than saying one batch to. Okay. Yeah. Okay.
Thank you.
So now I would like to introduce you all to session chair and co-chair, Dr. Vijay Chaudhary, ma'am. Dr. Vijay Chaudhary is the organizing chair of Conference 3 Sustainable Development in Applied Science, Engineering and Technology of the 4th International Convention on Green Environment, Technology and Entrepreneurship through Innovation. She is a research-oriented senior academician with an excellent track of more than 20 years of research and teaching experience in the field of microbial biotechnology. She was the only one person selected from all over India by Ministry of Human Resources Development, Government of India, and awarded the prestigious Canadian Commonwealth Scholarship in the year 2006 for pursuing graduate studies in biotechnology from Canada, which covered her entire expenditure of tuition fees, health insurance, traveling and living cost, books, laptop, etc. Her research work of fungal biotransformation under the guidance of Canada Research Chair and Tier 1 Professor, late Professor Thomas Hudelicki, was fully funded by Normaco Incorporated of the pharmaceutical giant Johnson & Johnson USA. She has many national and international publications to her credit and has been granted one Indian patent. She has also contributed to the university administration as a member of NAC committee and gender cell and alumni coordinator of Amity Institute of Biotechnology. She is one of the founding faculty of Amity University Biotechnology, Amity University, Rajasthan. Dr. Geeta Sanket Joshi, Associate Professor at Amity University of Microbial Technology. Dr. Geeta Sanket Joshi has 18 years of experience in teaching and research. She has specialized in areas as plant growth promoting rhizobacteria, environmental bioremediation, transgenic rhizobia, and microbial enhanced oil recovery. Her work has contributed significantly to advancement in these fields, demonstrating a strong understanding of both theoretical and practical aspects. From 2011 to 2022, she worked as a teaching consultant at Sultan Qaboos University, Oman, in the Department of Biology. Her research has earned a total of 1,390 citations, reflecting its impact in the academic community. With the H index of 13, her work shows influence and consistency, while an I-10 index of 14 highlights her contribution through widely cited publication. With this, I would like to end our today's oral presentation session. I would like to thank all the participants and our audience. Thank you.
जाओ ये बंद कर दूं साहब?
This is a notification for everyone that the buses are going to leave at 6 p.m. and we are going to start our cultural event in just 10 minutes. Thank you. Hello. Check. Hello.
Hello. Check.
Hello. Check. Hello. Check. Hello. Ladies and gentlemen,
सर वो लीव कर दो मीटिंग.
Hello. Check. 1, 2, 3. Hello. Check. Hello.
Hello. Check. Hello.
Hello.
मेरा वाला सही का?
Check.
Hello. Check. Hello.
चेक करने के लिए बुलाया सर.
Hello. Check. Hello.
माइक तो चल रहे, कहून भी करना है अभी सर. माइक तो चल ही रहे हैं, कहून पे तो माइक लगे.
बीच में अगर
Check.